בוקר טוב, אני שמח להציג את פרופסור תומאס מונטין מסטנפורד, and I'll switch to English now. So I'm very honored to present today Professor Thomas Montine from Stanford University to our staff meeting here in Rambam. Professor Montine holds a bachelor degree in chemistry from Columbia University, PhD in pharmacology from University of Rochester, and an MD from McGill University. His postgraduate medical training was in Duke University, and he was junior faculty in Vanderbilt University, where he was awarded the Thorne Professorship in Pathology. In 2002, he was appointed the Elwood Endowed Professor in Neuropathology and the Director of Division of Neuropathology in the University of Washington. He was Director of the University of Washington Alzheimer's Disease Research Center, and also the founding director of Pacific Udall Center in 2009. And this was one of the 9 NINDS funded, correct, by Maurice K. Udall Centers of Excellence for Parkinson's Disease Research. In 2010, Professor Montine was appointed chair of Department of Pathology in University of Washington. In 2016, he was appointed chair of the Department of Pathology at Stanford University and the Stanford Medicine Endowed Professor in Pathology. Professor Montine was the 2015 president of the American Association of Neurodegenerative Diseases, and he's currently the chair of the FDA Advisory Committee on Peripheral and Central Nervous System Drugs. Professor Montine's lab focuses on the molecular and biochemical basis of cognitive impairment and in aging and neurodegenerative diseases, with the goal of defining key pathogenic steps and thereby new therapeutic targets. Professor Montine is among the top recipients of NIH funding for all department pathology faculty in the United States, and in PubMed there are 749 publications of Professor Montine, and a total citations more than 97,000. That's very impressive. Professor Montine's talk today will be about the toward precision medicine in Parkinson's disease, and I'm very honored. Tom, thank you for coming, and the stage is yours.
Good morning, and thank you for an overly generous introduction. And while I have the microphone, I know I can speak for all of my colleagues from Stanford. Thank you so much for what seems like your endless generosity and hospitality. Thank you for inviting us back. I decided to speak today about collaborative work that we've started with Dr. Schlesinger's lab, and to focus on therapeutics that we're developing for Parkinson's disease. If you don't mind, I'll just keep going. If you can hear me in the back. If you can't, let me know, I'll speak up. Okay. See, Yvette will take care of this. So this is a photograph of Stanford's campus. Many of you have visited us. In the foreground is Hoover Tower, and then the buildings of our campus. The last building in the image is our new hospital, and then in the background are the Santa Cruz Mountains. Those are my disclosures. I don't have any financial conflicts with what I'll be discussing today, although 2 of the patents that are listed there cover the chemical matter that I'll be talking about later in the talk. So as was said, my lab focuses on neurodegenerative diseases. This is an image of the human hippocampus, the region of brain that makes memories. We took this image with a type of microscope that we built in our department called a MIBI, and I just think it's such a beautiful image. I'd like to start with this. Our patients ask us 2 questions, always 2 questions. What's wrong with me, doc? And can you treat it? Yesterday, we had a neuroscience roundtable, and I outlined some of the work that we do to try to answer the first question, What's wrong with me? We do a variety of population-based studies to drive a genetic analysis of endophenotypes related to neurodegenerative disease. Today, I'd like to talk about our work addressing this question: can you treat it? I'd like to talk about a collaboration that we've started with Dr. Schlesinger and her team around precision medicines for Parkinson's disease. These are the 6 topics that I'll touch on in the next about 40-45 minutes. So first, for those of you that are not neurologists or involved in neuroscience perhaps, what's Parkinson's disease? This is a famous photograph from the mid-1800s taken by a student of J.Montine Charcot's at l'Hôpital Salpêtrière in Paris. This man's first name is Pierre. His last name is lost to history. His last initial is D. This is Pierre D. And in this image, Charcot was capturing the essence of Parkinson's disease. What intrigued him was this disease that simultaneously causes too little movement and too much movement. It took us many decades to start to understand the different facets of this disease. One key feature is the loss of this molecule. This is dopamine. It's a decarboxylated form of a modified amino acid. It's a very simple molecule. It's used by dopaminergic neurons, of which there are about 80,000 in the human brain. So out of the tens or hundreds of billions of neurons in our brain, only 80,000 use this neurotransmitter, and they express widely throughout the brain with 3 major pathways. One pathway, shown here in blue, to the striatum, that's controlling movement, and so is responsible for the major motor features of Parkinson's disease. But the dopaminergic neurons also project to this region of brain called the nucleus accumbens, and that's where dopamine has its prominent impact on behavior, risk-taking behavior, for example, reward behavior. And then the third major projection of the dopaminergic system is to the frontal cortex, the region of brain that most distinguishes us from the great apes. And in all these regions, the dopamine, although it's called a neurotransmitter, is really a neuromodulator. Its release is fine-tuning the excitatory and inhibitory circuits that are running through these different regions of brain. The other major facet of Parkinson's disease is the accumulation of an abnormal form of a protein that we all are now expressing at high levels in the neurons in our brains, a protein called alpha-synuclein. Somehow, and it's yet unclear how, the alpha-synuclein adopts an abnormal shape, or it's misfolded, and then aggregates into these structures that are called Lewy bodies. So those are the 2 key features of a disease that remains somewhat enigmatic. So Parkinson's disease clearly has a motor component, but lest we forget, quite debilitating to patients is also the cognitive and behavioral impairments that accompany the disease. And then these 2 pathologic features, the loss of dopaminergic neurons and the accumulation of this pathologic form of alpha-synuclein. And you'll notice, this will be a theme that we go through today, this was the view of the disease, I'd say, as recently as 15 years ago, maybe 20 years ago, very neuron-centric, right? Neurons are lost while they're accumulating alpha-synuclein. I think now we understand that the cell biology of the disease is much more complicated than that. So that is a brief introduction to Parkinson's disease. We need to spend a little time understanding the genetics. And so this is work that's developed over the last 20 to 25 years. It's important to understand that human genetics defines the actor, but not the role. So to use that metaphor, it'd be like genetics would tell us that Sir Laurence Olivier is starring in a production. That's all it would tell us. It doesn't tell us if he's singing or dancing. It doesn't tell us if he's the lead actor or a bit player. It doesn't tell us if it's a film or a theatrical performance. All we know is that he's somehow important, right? So that's what genetics tells us. Who are the actors? So this is a summary of the major genetic contributors to Parkinson's disease. Currently, the graph is divided, spread out on two axes. So on the y-axis is the risk for Parkinson's disease, either low, medium, or high risk. And then there's this fourth group that's causal. And that's, in genetic terms, causal means that if you inherit this particular variant and this particular gene, your risk of not developing Parkinson's disease is 1 in a million or less. So you're almost certainly going to develop the disease. That's what causal means. So I'll draw your attention to 3 of these genes. The first one is SNCA. That's the symbol for the gene that encodes alpha-synuclein, the protein I was just telling you about that becomes misfolded and aggregates in Parkinson's disease. So there's very strong pathologic, biochemical, and genetic evidence that alpha-synuclein is important in this disease. There are many labs around the world that are trying to develop therapies that address this abnormal alpha-synuclein that accumulates in brain. Another gene I'll call your attention to is GBA. This is the gene that encodes for glucocerebrosidase. If an individual inherits 2 mutant copies of GBA, that's the basis of Gaucher's disease, a visceral and neurodegenerative disease of children and young adults. Inheriting a single mutant copy of the GBA mutation is a common contributor to Parkinson's disease, and so there are efforts underway to develop therapies directed at that target. And then I'll draw your attention to this last gene, LRRK2, the leucine-rich repeat kinase 2, LRRK2, is present in all 3 groups. So there are disease-causing mutations in LRRK2. There are considerably more common diseases that substantially raise your risk, 5 to 10-fold for developing Parkinson's disease, and then there are also these low-risk genes. The focus of what I'll talk about today is on LRRK2. LRRK2 was discovered as a genetic contributor to Parkinson's disease, and then quickly after that was recognized as also contributing to the risk for Crohn's disease and leprosy. Variants in the LRRK2 gene increase risk for Crohn's disease. They decrease risk for Hansen's disease, a form of leprosy, and other variants yet in the gene contribute either positively or negatively to the risk of some forms of cancer. And this was a bit of a head scratcher when it was first identified, right? Because back then, our concept of Parkinson's disease was a disease of neurons, and these are not diseases of neurons. In fact, these are diseases that we consider inflammatory or major contributors from the inflammatory reaction. And that helped evolve our thinking of this disease to one that, yes, ultimately the neurons are degenerating, but perhaps the immune response is key to mediating that degeneration. So a little bit more about LRRK2. It's located near the centromere on human chromosome 12. And this is a map of the very large gene, 52 exons, and the massive protein, over 2,500 amino acids. Multiple domains. It's an unusual protein. It's not that it's an enzyme, but that it has 2 enzymatic sites, a GTPase in the ROC domain, and then a kinase, shown here in green. And we're going to focus the talk on the kinase domain of LRRK2. One of the mutations, in fact, the most common mutation in LRRK2 changes the amino acid from a G to an S at position 2019, so I'll refer to that as the G2019S mutation. It's in the kinase domain, and this is a map of the prevalence of the G2019S mutation in individuals who have Parkinson's disease, and you can see readily, so it's color-coded, right, from light colors to dark colors, dark being more prevalent. There's a very strong founder effect for the G2019S mutation in the Mediterranean region, so North Africa, the Middle East, and Southern Europe, where in those regions the prevalence of this mutation among Parkinson's disease patients varies from 20 to up to 40%. There's a paper from movement disorders clinics in Israel back a few years ago putting this number at just below 40% for the patients with Parkinson's disease in your country. It's much less common in the United States, and this just has to do, right, there's such a strong founder effect from the Mediterranean region. It just has to do with who has immigrated where in what numbers, dictates the prevalence around the rest of the world. So LRRK2 variants, they're the most common genetic risk for Parkinson's disease. The G2019S mutation is the most common variant in LRRK2, and there's this very strong founder effect in the Mediterranean. If genetics defines the actor, then mechanism defines the role. So to use that metaphor again, now we're not going to learn that it's simply Laurence Olivier who's starring, but we're going to learn that he's the lead actor who's singing in a Broadway production, right? So genetics versus mechanism. So what does LRRK2 do? This is a rendering of the structure of a dimer of the LRRK2 enzyme, and highlighted in purple are the kinase domains. A kinase is a protein, an enzyme that catalyzes the reaction of moving a phosphate group from ATP to another protein. So that's what's diagrammed here. So here's the substrate protein. The kinase that we're talking about is LRRK2, and it moves a phosphate group from ATP to the substrate protein. Right? This is a fundamental mechanism by which cells signal inside the cell. In fact, there are over 500 human kinases occupying a large amount of the human genome. So this is a critically important process for controlling the intracellular activity of cells. Excuse me, let me get restarted. Excuse me for that. I hope that won't happen again. So here we were. So that's what LRRK2 does. What does the mutant LRRK2, not just any mutant LRRK2, what does G2019S LRRK2 do? Does it have a different mechanism? It doesn't. It doesn't have a novel mechanism, but its kinase activity is increased 5 to 10-fold. So the G2019S LRRK2 mutation is a gain of function, and it's a hyperactive kinase. So it's too active. The cell biology of LRRK2 is quite complicated. It sits at the hub of a signaling network that influences many, if not all, intracellular processes. They're grouped here in this diagram as organelles and microtubular structures, but you look within that circle, and this kinase activity of LRRK2 has been linked to just about every type of process that occurs within a cell. I draw your attention to one, because we'll come back to it later, a nuclear factor, kappa B, NF-kappa B, but that's just one of the many activities that LRRK2 kinase influences. So that's what it does, and then where is it? It's expressed predominantly in this cell type, the monocyte and tissue-resident macrophages that exist in all organs, including brain, and in brain those cells are called microglia. And then in other organs, it's expressed to high levels in type 2 pneumocytes. Those are the cells in lung that produce surfactant. It's also expressed at high levels in kidney, and that's going to become important in just a few moments. So the answer to this first question, what's wrong with me? Well, if you are unfortunate enough to inherit this bad actor of G2019S LRRK2, what's wrong is that the kinase activity is hyperactive, and it's hyperactive in immune cells, lung cells, and kidney cells. And you'll notice I didn't put neurons. Now, the field has engineered lots of different neurons to express LRRK2, but under normal regulatory circumstances, LRRK2 is expressed at undetectable levels. So I can't say there's none in neurons, but it's undetectable in neurons. It's detectable in microglia in brain, the tissue-resident monocytes in brain. But we published this some years ago in Nature Genetics. We can identify it in the microglia in brain, but not in neurons in brain. So I can't say it's not there, but if it's there, there's very, very little. So then to move to therapeutics. An overview of first and second generation therapeutics first. So this is a diagram, again, of LRRK2 laying out the kinase domain here, and then this rendering of the structure of the enzyme. Here are, inside these two boxes, the first generation and then our attempt at making second generation LRRK2 kinase inhibitors. So the two that I'm showing you, MLE2 and DNL201, these are first generation inhibitors. And what defines a first generation inhibitor, that it potently inhibits LRRK2 kinase, but it inhibits both the wild type kinase and the mutant kinase with the same potency. There's no discrimination between wild type and mutant. And that's, I think, a fundamentally important point, because the human genetics tells us that it's the mutant LRRK2 kinase that's important in Parkinson's disease. It does not tell us that wild type LRRK2 kinase is important in Parkinson's disease. So these are non-selective. They'll inhibit both types, and as you'll see in a moment, generate some concerns about toxicity. So our entry into this field about 5 years ago was to try to develop kinase inhibitors that were specific to the G2019S mutation alone and would not inhibit the wild type form of the enzyme. And we thought that might be important, as I said, because the rationale for this target, at least the human genetic rationale, is that the mutant form needs to be inhibited, not that the wild type form needs to be inhibited. And as I said, there are concerns about on-target toxicity, meaning there's concerns about the non-selective inhibitors by virtue of inhibiting the wild type enzyme in lung and kidney, causing some toxicity. This is a surface rendering of the enzyme, and the magnifying glass is showing one of our type 2 inhibitors embedded in the active site. It's this compound, compound 3. I will refer to the series that we've invented as either, there are two series, an indazole and a pyrazole. Those are names of chemical structures. So just to orient you, this is an indazole, right? The same structure as ATP. So ATP is the natural substrate for the active site. So we're trying to mimic that when we build these chemicals. You can see there's also an indazole in MLE2, and that indazole is laying right here in, as we envision it sitting in the active site. And what we have done, basically, is start with, in our indazole series, start with an indazole, then see MLE2 doesn't build much in this direction. We have built quite extensively off in this direction of the indazole. And the rationale for that is we're reaching from the indazole here out across the active site to engage directly with the S of the G2019S mutation. That's the logic. So can we treat it? That's the second question. Well, we're trying to, right? So we're developing small molecule inhibitors. We and many others are developing small molecule inhibitors of the kinase in LRRK2, right? That's our target. And the difference between what we are doing and what others have done is we're making ours to be selective for this mutation in the hope that we'll avert on-target toxicity. I will say that in my part of the world, if we're completely successful, that we will have created an orphan drug. There'll be less than 200,000 people in the United States for whom this is indicated. In your part of the world, it would not be an orphan drug, right? Because somewhere between 30 and 40% of your patients with Parkinson's disease have this mutation. So we put together a movie for you. I hope this runs. There we go. So that's a surface rendering of the enzyme. It's going to change every 8 seconds, so give it a moment. This is the same image. Now the enzyme has been converted to the arrows and ribbons, and that's ATP sitting in the active site. And this is one of our pyrazole compounds sitting in the active site like ATP and then reaching across to the G2019S. That S is right over here. And then this is one of our indazole series. There's the indazole, and again, we build it out to bring this nitrile out over by the serine, the mutant serine. And then we go back to the original image. These two compounds from Genentech, GNE, now taken by Biogen, are in clinical trials. So far, phase 1 trial showed that it was tolerated without apparent acute toxicity. There was a paper published last week, with these two compounds given to patients and healthy volunteers, patients with Parkinson's disease and healthy volunteers for 28 days. And over that 28-day period, there was no apparent toxicity. So that's good. We don't know anything about effectiveness yet in the trials. That's yet to come and is predicted to be 2 or 3 years away. 28 days is not a very long exposure for a drug that, if it is effective and given to patients with Parkinson's disease, will be taken every day for, could be up to 20 years, right? So there's no acute toxicity. Why do I keep referring to concerns about toxicity? I mentioned earlier that wild-type LRRK2, which is targeted by these two drugs, wild-type LRRK2 is expressed at high level in lung and kidney, and I'll show you some data, some of the data that has raised concerns about this. So this is these are data from an experiment in mice where we're not using any drugs now. We've removed LRRK2 genetically. So these mice express no LRRK2. And if you don't do anything to those mice, just let them mature and get older, these are photomicrographs of lung. And the key here is that the empty spaces, those are the alveolar sacs, that's where gas exchange is happening. There's a loss of alveolar spaces from fibrosis of the lung in the absence of LRRK2 in an adult mouse. And if you just follow across, then if you challenge the mouse with bleomycin, that presents a free radical challenge in lung, meant to model conditions like if the individual smokes or has asthma, then the fibrosis of the lung is really quite prominent. And so this is one piece of evidence that raises concerns about chronically suppressing wild-type LRRK2 activity. Then another group has used one of these two compounds that I'm showing you in non-human primates. And these were experiments that went on, as I recall, I could be wrong about this, approximately 3 months. So again, not anywhere near the length of time that we would be expecting patients to take these drugs. And you can clearly demonstrate these early changes in the type 2 pneumocytes, the cells that naturally express high levels of this enzyme. They accumulate these abnormal structures within their lungs. So there's these pathologic changes in both mice and non-human primates when LRRK2 is continuously suppressed, either genetically or by one of these compounds. So it's a major milestone to have created these compounds and move them to clinical trial. They're potent. We don't yet know if they're effective, but they're potent. So they get to target, they do what they're supposed to do. They get to target, they inhibit target. They're non-selective, and we think that's a gap that we can improve upon. And they do produce, at least in mice and non-human primates, they produce lung toxicity. And so the question is, what will that mean for older individuals who take these, who chronically suppress wild type like LRRK2 with one of these compounds for years or decades? Okay, so then what type 2 inhibitors have we made? For comparison, I'm showing you MLE2 again, one of the first generation non-selective inhibitors. Then in the table that runs underneath is potency and selectivity. So potency is how powerfully do these compounds suppress kinase activity. And that's measured by what's called the EC50, so the effective concentration that produces a 50% reduction in kinase activity. So these are EC50 values. And successful kinase inhibitors are usually in low nanomolar concentrations. So like MLE2 is 30 nanomolar. A number that low is targeted, right? So that's parts per billion. The reason we target such a low number, as I said, there are over 500 kinases in our cells, and we only want to inhibit one of them, right? Not any of the other ones. And so once you start drifting up to higher concentrations, you have non-selective activity within the kinome. So MLE2 is potent, 30 nanomolar. It's non-selective. It's almost unity, meaning we're comparing the activity in wild type versus G2019S LRRK2 kinase. Then two of our compounds. One, an example from the indazole series. This was the compound 3 that I showed you before. And just to give credit to the chemists that do all the work, we don't make you don't get to the solution in 3 compounds. It takes many, many iterations to get there. So its real name is compound 3322. So 3322 is just like the compound 3 I showed you before. And so to interpret these graphs, first look at the action of MLE2 in wild type LRRK2 kinase and G2019S mutant LRRK2 kinase. And these are expressed in human cells. And so the blue and the red curves overlap each other, meaning there's no discrimination. It's inhibiting wild type and mutant with the same potency. And then our compound, 3322, against wild type LRRK2 is in green, and against the GS mutation is in green, and you see how the curves diverge. So we inhibit the mutant at a much lower concentration than we do the wild type form of LRRK2. We're not quite as potent as MLE2, as you can see here. The EC50 for 3322 is 40 nanomolar, but we're 2,500-fold selective for the mutant. Right? So this is our best compound in the indazole series, partly because of these parameters, also because it's brain penetrant, at least in mice, and can engage target in brain. So we're going forward with 3322 as our lead compound in the indazole series. And then this is an image of compound 4649. This is on the pyrazole scaffold, so it's hard to see, but there's the pyrazole embedded there in the active site. The pyrazole is just a simplified version of the indazole, and it gives us greater flexibility in the medicinal chemistry. And again, you can see we've built out the molecule to bring the sulfonamide group, this group, all the way out to engage with the mutant S, the mutant serine amino acid. And these are the performance characteristics of this inhibitor, so I won't belabor this, but you can again see that MLE2 has no specificity, right, for wild type versus mutant LRRK2. 4649 is actually more potent than MLE2, slightly, at 16 nanomolar EC50, so even a lower concentration achieves 50% inhibition. And then there's this really striking difference in activity between mutant LRRK2 and wild type LRRK2 for 4649, 6,200-fold difference in activity between the wild type and the mutant enzyme. So the second generation inhibitors that we've been able to make, they're as potent as first generation. They're highly selective for the G2019S, and because of that, we're hypothesizing that they will avert this on-target toxicity in the lung and potentially in the kidney. So with all of that as a setup, I visited here last July and met Dr. Schlesinger and described what we were doing, and she said, oh, well, we can help. The next step at these experiments is to use the compounds in human cells that actually carry the mutation. Everything that I've showed you up to this point are cells or enzymes that we have somehow genetically engineered, right? What cell type do we focus on? The major cell that expresses this enzyme is monocytes and tissue macrophages. Monocytes are accessible through peripheral blood, and so Dr. Schlesinger and her team have drawn blood and prepared the white cells in blood from individuals with these different LRRK2 genotypes. So individuals with Parkinson's disease who have both alleles of LRRK2, right? You get one copy from your mother and one from your father. So both copies, they have Parkinson's disease, but both copies of LRRK2 are normal. So they have Parkinson's disease for another reason, not because of LRRK2. Then individuals who have one normal copy and one mutant copy of LRRK2. And then there was one person who inherited a mutant copy of G2019S LRRK2, both, one from his or her mother and the other from their father. Okay? So the advantage of this, they're human cells. We haven't genetically engineered them, so we don't think we're introducing spurious modifications, but there's no chance to introduce a spurious modification. We haven't manipulated these cells. We used a technique that was pioneered at Stanford called CyTOF. I learned yesterday that your campus has a CyTOF machine. They are amazing. This is the 40-plex panel, so we're probing. Sorry, I'll pause and take a step back. So CyTOF is a flow cytometer, meaning that individual cells are isolated as they come through the flow cell, one cell at a time. Each time that cell comes through, it's atomized, and the antibodies that have been tagged with heavy metals then are also atomized, swept into the mass spectrometer, and the mass spectrometer reads out what heavy metals came through on that cell, right? So it's the heavy metals are the reporter, the antibodies engage the targets, and we're looking at one cell at a time, 40 endpoints on each cell. So millions of cells come through, we collect these 40 endpoints on each one of them. And then we can render this sort of map. This is, right, so how can a human comprehend 40-dimensional data, right? You can't. So there are what are called dimensionality reduction techniques. This is one of them called UMAP. So we've taken these 40 data points and reduced them down to a 2D graph. And it's color-coded for the different types of cells that are in blood. So there are CD8 positive T cells, CD4 positive T cells, natural killer cells, B cells, granulocytes, and myeloid cells, another name for monocytes. They're in purple. These are the monocytes here. So this is the first time this has been attempted with blood from individuals with LRRK2 mutation. All previous data is just from blood cells, which is all these cells mixed together, which is fine, but imprecise. The LRRK2 is expressed at high level in the monocytes. So we want to focus on the monocytes. The rest of the cells, they're just a distraction, and they're going to dilute whatever signal we might detect. So all the data I'm going to show you is from the purple cells, the monocytes only. Then if we've laid out these graphs, I should pause and say, please take this the right way. So we decided we were going to collaborate together last July. It took until the first week of December for the lawyers at Stanford and the lawyers at Rambam to agree that we could exchange these cells properly. And so we've done this experiment twice. So 7 months, 5 of it spent on just getting the shipment, but we've started working with them. So this is just our first CyTOF experiment. So the way I've laid out the results for you is we're now going to compare individuals, monocytes, who have, everyone has Parkinson's disease, individuals who have normal LRRK2 versus one mutant copy of LRRK2, and we're looking at the change between those two groups. So we're using these individuals to control for each other, and they're color-coded from no change in blue to high change in red, and then we're looking at 4 key intracellular signaling proteins, S6, CREB, I-kappa B alpha. So I drew your attention before to NF-kappa B. I-kappa B alpha is its partner. NF-kappa B modulates, it's a powerful modulator of gene expression, and I-kappa B alpha is the brake on NF-kappa B. And then the last is P38. So when we compare unstimulated wild type to heterozygous G2019S monocytes, there's no difference in the expression of these 4 key intracellular proteins. When we stimulate the monocytes with lipopolysaccharides, so this is a lipoprotein expressed on gram-negative bacteria, and when monocytes encounter lipopolysaccharide, they go berserk. So these are highly, highly activated, stressed, if you will, monocytes, and the wild type and mutant behave the same. We then introduce our selective inhibitor into these stimulated monocytes, and now we begin to see differences. The difference resides at expression of I-kappa B alpha, which, if the way the ratios go, it's red on this graph, so it's very high in the wild type cells, and we've suppressed it in the mutant cells. So our mutant, what we believe is a mutant selective inhibitor, has selectively inhibited the increased expression of I-kappa B alpha in these patient-derived cells. And then the last part of this experiment, we used a non-selective inhibitor, and we don't see that effect at I-kappa B alpha. So this is what we were slapping each other on the back. This is exactly what we were hoping to find. Selective activity of the second-generation inhibitors in the mutant-containing cells. That's what we were hoping to find. We did. So then we did the next experiment where, same experiment, now we're comparing the wild-type cells to the one cell type that had both LRRK2 copies mutated, the one sample, one patient, and we see essentially the same thing. Right? There are weaker signals, especially around P38, that we will follow up, but the big signal is at I-kappa B alpha, large and reproducible. And then we compare people who had one mutation to the person that had 2 mutations, and this is called the gene dosage effect. So there appears to be a difference in the outcome of this experiment depending on how many mutant copies you have. And again, we have this specific reproducible effect that we're seeing with the second-generation inhibitors. So thank you so much. This experiment is impossible, virtually impossible for us to do without the partnership. So thank you again. That's a diagram of what I-kappa B alpha does, but I think it's enough to say that it's a brake on NF-kappa B. So this critical experiment shows that our type 2 inhibitors selectively act on the G2019S carrier cells, and in it they're suppressing the induction of I-kappa B alpha, a key regulator of the immune response. So to summarize then, Parkinson's disease, it's a neurodegenerative disease with motor, behavioral, and cognitive impairments. All of them are important to our patients. We've focused on G2019S LRRK2 because it's the most common genetic contributor to Parkinson's disease, and it's a gain of function mutation, which is great when trying to build a small molecule intervention, and it's a change from a glycine to a serine. So if there are any biochemists in the room, you immediately recognize that that's an exploitable change. So G2019S is a key actor in Parkinson's disease. The role that it plays is as a hyperactive kinase, and its performance is mostly in immune cells. The first generation LRRK2 kinase inhibitors are a major milestone as the field moves forward. They're potent, but they're non-selective, and they cause on-target toxicity in the lung in mice and non-human primates, and it's a question mark what that will mean for older individuals who need to take these compounds for years and years. So that's, as I say, that's the gap we're trying to step into, to be prepared with more precise interventions. And then finally, we've invented these second-generation LRRK2 kinase inhibitors. They're as potent as the first generation. Their difference is they're highly, highly selective for the G2019S mutation. And then our hypothesis is that this will avert the on-target toxicity, and those are experiments we're doing now, long-term exposure in mice to see if we avert the toxicity. We shall see. So I need to thank the people that do the work. This is a picture of the scientists that work in my group from last summer, the people who fund us, the Farmer Family Foundation, and then of course the NIH. And then to thank you again for the invitation to speak, and you're making us feel so welcome in your home. Thank you.
Thank you. Thank you very much.
My pleasure.
For a very interesting lecture. The session is open for discussion, questions. Professor Skoretsky.
Since it's a gain of function, or Laurence Olivier is overacting, is RNA medicine another approach you're following to try to decrease?
Others are. I have no expertise in developing.
Would it make sense? Would it make sense to?
Yes, it does, totally. To my logic, any platform that can selectively reduce the mutant form of the enzyme would be worth pursuing.
So that's my question. Since you have to be selective for the mutant form, would you be reducing the entire? Can you do it selectively for the mutant form with exon skipping, or you could?
Yeah, I think so.
And you emphasize the lung toxicity, but you said the now state is also renal toxic. Yeah. Is that not a concern?
Well, it's a developmental abnormality. It is a concern for the mice. It's a developmental abnormality. It's a developmental abnormality in the kidney in the mouse, but the drug wouldn't be used in people until they're well past developmental stage. Yeah.
Yeah. How prevalent is this specific gene mutations among Parkinson patients? I mean, how many of the patients with Parkinson such a therapy can be relevant?
Right. So it varies in what part of the world. In the United States, it's somewhere between 1% and 2% of people with Parkinson's disease. In this part of the world, what's published is it's somewhere between 30 and 40%. And that's because of the founder effect, right? This mutation arose somewhere in the Mediterranean region around the 13th century. So the mutation is quite prevalent in this, most prevalent in this region of the world, but the expert is here. North Africa also, and Southern Europe, but here in the Middle East, and as you say, in some cities in the United States, quite prevalent. So it's a precision medicine, right, which I think the future of, well, let's say this path to the future of medicine will require an ensemble of therapeutics that have different precise molecular targets, so that I imagine a future conversation is someone presents with Parkinson's disease, they're determined to have this mutation, they would receive drugs like this. If they were determined to have a GBA mutation, they would receive a different therapeutic. If they had no identifiable mutation, yet another therapeutic approach. I think that's the future.
Okay, one note in Hebrew. בשבוע הבא תינתן הרצאה על ידי פרופסור מרסל מחלוף, ראש המעבדה למתן תרופות לסרטן וטכנולוגיות מבוססות תאים בפקולטה להנדסה ביוטכנולוגית ומזון בטכניון. נושא הרצאתה רוח רפאים, מערכת הובלה של תרופות או תרפיה. פרופסור אלברט טל, please.
So, first, once again, all of you for coming here and for this continuous collaboration. I think, you know, these kind of things, this is exactly what we are talking about. And I'm sure that there's much more options to do more stuff, and we are waiting for that. It's intriguing couple of days and for both of us. So once again, thank you very much. And we thank the vision of Lloyd that, you know, started all this with Rafi 7 years ago, something like this. So we are extremely excited around it. So, well, whoever gives a lecture at Rambam gets this. I'm sorry about the overweight that you'll be charged for. And again, this is another Israeli invention, so if you have any problem crushing one of your genes, it would help. So please.
Thank you so much. Thank you. That's lovely. Thank you. Yeah. Thank you, everyone.